Welcome to this explainer. To kick things off today, I want you to picture a bright ninth grade biology classroom. You know the vibe. You're sitting at a neat study desk, sunlight streaming in, watching your teacher map things out on the whiteboard, and suddenly, bam, that complex concept just clicks. Everything makes sense. Well, today, we are going to channel that exact focused energy. We're looking past the textbooks to uncover how scientists actually crack the cases of the natural world.
Because, let's be honest, when you look at the sheer complexity of life, from a massive forest ecosystem down to the tiniest cell, it can kind of seem like magic, right? But how do scientists figure out how the whole shebang works? Well, instead of just guessing, biologists use a highly rigorous, step-by-step toolkit. They act exactly like detectives at a crime scene, following a strict biological method to make sure their findings are absolute, undeniable facts.
Okay, so here's our roadmap for today. We'll be moving through 5 core phases: spotting the scientific problem, making and categorizing observations, forming a hypothesis and logical deductions, running the crucial experiment, and finally, summarizing and reporting results.
And here is that foundational blueprint. Every single biological discovery you've ever learned about has passed through this strict sequence: recognition of a problem, observation, hypothesis, deduction, experiments, and results. It's a gauntlet, really, that turns simple, everyday curiosities into established scientific facts. Let's tackle these step by step.
Every great investigation starts with a trigger. So, step 1, spotting the scientific problem.
Science doesn't just happen in a vacuum. The very first step involves identifying and defining a specific issue or phenomenon that you actually want to investigate. This usually happens in one of two ways. Either a biologist just happens to notice something unusual while they're working, or someone else brings a specific, puzzling question to their attention. It's the initial spark that ignites the whole process.
To put that into perspective, imagine a biologist is out in the field and notices a patch of plants growing way taller than usual. The environment looks identical to everything around it, the weather is exactly the same, but the growth is undeniably different. Boom! They immediately have a scientific problem. What factors are responsible for the increased growth of these plants? They've just found their first mystery to solve.
Once we have our mystery, it's time for phase 2, making and categorizing observations. Time to gather our clues.
So, with the problem identified, scientists have to look incredibly closely at what's going on. They use their senses: sight, hearing, touch, smell. Actually, scratch that. Skip taste. Licking unknown biological samples is just a terrible idea. But they also dig into previous research, and they don't just throw all this info into a messy pile. They categorize these clues into 2 very distinct buckets: qualitative and quantitative. Qualitative observations are things you can't really measure with numbers, like the vibrant color or smooth texture of a flower. Quantitative observations, on the other hand, are all about hard numbers, like counting exactly 5 birds in a tree.
Now, the absolutely crucial point here is that quantitative data is considered far more accurate. Why? Well, because qualitative descriptions can be super subjective. What looks bright red to me might look kind of orange to you. But quantitative observations, they are invariable. They are measurable. Cold, hard numbers leave literally zero room for misinterpretation or bias. 5 birds will always be 5 birds.
All right, now that we have our carefully categorized clues, we enter hypothesis and logical deductions. This is where we form a theory.
Based on all those observations, scientists develop a statement that might prove to be the answer. We call this tentative answer a hypothesis. But listen, a hypothesis isn't just throwing random guesses at the wall to see what sticks. To be scientifically valid, it must strictly adhere to 4 rules. It has to be a proposed answer to the problem. It absolutely must match your available observations. It has to be testable, and crucially, this is the big one, there must be a way to disprove it. If you can't test it and potentially prove it wrong, it's simply not a scientific hypothesis.
Let's look at a real-world hypothesis straight out of the biology playbook. Leaf discoloration and stunted growth in a plant are caused by a deficiency of iron in the soil. This hits the nail on the head. It proposes an answer we can test, it lines up perfectly with what the biologist observed, and most importantly, we can easily test the soil's iron levels to see if this idea actually holds up. Once they have that hypothesis, scientists draw logical results from it, which we call deductions. They use this brilliant if-then logical pattern. For instance, if iron deficiency is causing the symptoms, then adding iron to the soil will lessen the leaf discoloration and promote healthier plant growth. It's essentially predicting exactly what's going to happen if the hypothesis is true, which sets the stage perfectly for our next step.
With our if-then deduction firmly in hand, it is time for running the crucial experiment, the ultimate test.
This is where the rubber meets the road. Scientists perform experiments to test all their hypotheses. This is the crucible. And here's the thing, in a successful experiment, it's not just about proving you're right, it's about proving everything else wrong. The incorrect hypotheses are ruthlessly rejected, and the one that actually withstands all the testing is finally accepted. From there, scientists can make new deductions and run even more experiments just to absolutely confirm they got it right.
To make sure their experiments are completely flawless, scientists arrange two identical settings. Take a classic example, testing if carbon dioxide is necessary for photosynthesis. They set up two similar plants. One is the experimental group. This one is actively deprived of carbon dioxide. The other is the control group, and it gets carbon dioxide just like normal. By creating this stark contrast, they can clearly see that photosynthesis stops in the experimental group, but keeps going in the control group. That control group proves that the change was strictly due to the missing carbon dioxide, perfectly validating the hypothesis.
But a detective's job isn't finished until the paperwork is filed, right? Let's move to our final phase, summarizing and reporting results. After all the experiments are done, biologists have to carefully compile their data. You definitely can't just hand in a coffee-stained notebook full of random scribbles. They have to use precise statistical analyses and create summarizing graphs to make the results crystal clear. Plus, they are required to include a list of references to acknowledge any outside information they used. Integrity and clarity are absolutely paramount here.
Ultimately, science is a massive team sport. These hard-earned findings are pretty much useless if they're kept a secret. So, scientists publish their results in journals and books. They share their findings at national and international meetings and in seminars. This ensures that researchers all over the globe can learn from the solved mystery, double-check the work, and build upon it for the next big discovery.
And there you have it, the exact structured method that takes us from a simple passing curiosity to a published scientific fact. So, the next time you face a complex question or just notice something kind of unusual in your everyday life, remember this method. Don't just guess. Categorize your observations, build a testable hypothesis, and ask yourself, what is the very first if-then deduction I can use to test my way to the truth? Thanks for joining me for this explainer, and keep questioning the world around you.